Hear me very well? Okay, great. So today he's going to talk about re-imaging gene environment interaction in the omnigenetic era. Let's welcome Church.
Yeah, thanks a lot. There will be some difficulty because of the Zoom issue, but definitely let me know if anything is unclear. I think you can at least put your questions into chat, and I can see the questions, but Jiwei, I assume, will be able to tell me if anything comes up. Obviously, as you can see from the title, I'm going to talk about gene-environment interactions. This is something that we started working on relatively recently. But my goal for today is I'm going to have a few slides for background stuff, and then specifically talk about one project that we have wrapped up. But then in the later half, I'm going to briefly talk about some very exciting work that is about to come out. So that's actually work in progress, but with a very high completion rate. But the overall point of this doing this seminar at my own department, I think it's partly to let everybody know that what we are actually working on. So I'll try to make sure the topic is broader than just the title, and give you a rough sense about some of the other exciting work we have been working with. And as I said earlier, let me know, interrupt me if anything is unclear. Anyway, so just a little bit of background, make sure we're on the same slide. We work on genome-wide association study quite a bit. And the idea of genome-wide association study is not new, and it's statistically pretty boring, actually. The idea is you study these single nucleotide polymorphisms. By the way, I assume you can't see the mouse, which is a bummer. So I'm going to try to describe where I'm at on the slide. But on the top right corner, you can see this genetic variation, which is a single nucleotide polymorphism. These are genetic variations that your DNA could differ from mine. And it's simply a type of variant with a single substitution of a base pair in the genome. What we do in GWAS, or genome-wide association study, is we want to study whether any of these variants in the genome is associated with a phenotype. It could be a quantitative phenotype like blood pressure or height. It could be a disease outcome like Alzheimer's or autism. And basically, we can do a case control study and compare people's DNA. And after doing GWAS, what you can get is something like the lower right corner, which is a Manhattan plot. The x-axis will be where each genetic variant is on the genome, and the y-axis will be the transformed p-value that quantifies the association between each SNP and the outcome of interest. You can see peaks on the Manhattan plot that will hint at genomic regions that actually contribute to the risk of the disease. Nothing special, and one thing to note is, in GWAS, we're actually doing marginal regression analysis. There can be a bunch of covariates, but the association really quantifies the relationship between a single variant and the outcome of interest. Then, one unique thing about GWAS in these days is sample size, obviously, is very important in almost all statistical problems, but it's actually often impossible to share these individual level rectangular data set that a lot of people will take for granted in other statistical applications. So whatever statistics you work on, you start with this n by p matrix with n individuals and p variables. But that turns out to be a luxury in human genetics research because of a lot of policy and privacy and just the politics in general. So people somehow are unwilling to share these individual level genotype data. What we can share, what people can share, is something like the lower right corner, which we call GWAS summary statistics. That's essentially the outcome of a GWAS, where you have finished running the association analysis between each SNP and the outcome. You get marginal regression coefficient. Like you can see in the figure, each row is a SNP. You have the SNP ID. You can see which chromosome it's on. You can see the location, the coordinate, and you have marginal regression coefficients. Like beta, you have standard error, you have confidence interval, and you have p-value. And you have 10 million rows like this, one for each SNP. If you are able to share this, then let's say we have a GWAS consortium. Someone can pull together these association statistics across 50, 100 cohorts that all contributed data like this and perform a meta-analysis, which is similar to what I'm showing on the left-hand side. Where basically, you have many, many different sites. Each site gives you the point estimate and the standard error. You can combine them through meta-analysis techniques and eventually boost your statistical power as if you're actually conducting a big GWAS after pulling together all the samples you have. So this is interesting because even if you are an author of this study, in a lot of cases, you still do not have access to the total data set you have. all you have, even if you're an author, is also something like the right-hand side, which is the summary statistics. This is the type of data we have to deal with in this field. And, but this actually effectively allows GWAS sample size to increase very quickly, because people are pretty willing to share summary statistics. Everybody does it. It has become a convention in the field. So the summary stats are now typically available. Looking at the left-hand side as an example, for example, early on, the largest GWAS for schizophrenia has a sample size of 7,000. People were not able to identify any genomic region associated to that. But when you have the sample size of 50,000, you identify 5 genomic regions contributing to schizophrenia risk. When you have 60,000, you identify 13 more loci. When you have 150,000, people identified 108 genomic regions contributing to schizophrenia. And on the right-hand side, you can see this is the latest preprint for schizophrenia GWAS. Now they have nearly 300,000 people, and in total identified 270 genome-wide significant hits, as you can tell from the Manhattan plot. And this is a pretty consistent observation for no matter what complex trait you want to study, which is that we know that for most human complex phenotype, we are talking about an extremely polygenic genetic architecture, which means that many genomic regions independently contribute to the same phenotype, but each of them only has a very weak effect size. So some people have coined this as omnigenic genetic architecture. I haven't seen people talking about this in the literature, but I thought that there's sort of 2 pillars in modern human statistical genetics research. One is that if you want to do fancier statistical modeling, your input data has to be something like this, which is GWAS summary statistics. And another special thing about statistical genetic research, or another pillar, is that your model has to be able to account for this extreme polygenicity or omnigenicity. You have to be able to deal with extremely large number of non-zero but very weak effects. Hopefully this can also be somewhat reflected by my specific projects later. One thing to note, though, is pretty interestingly, environment is almost always nowhere to be found in these studies. Some of this has, you can have an argument for some of this. For example, some people would think that whatever environment you're thinking of, it's not going to confound your genetic effect, right? Because obviously we don't want your genetic association to be confounded, but the reason why the genome-wide association study is somehow robust to such confounding effect is because we're talking about germline DNA variations that you get after you're conceived, certainly after you're born. These things don't change just because you eat or drink certain things or you go to certain school. So environment is not going to causally change your germline DNA variation. So because of that, in some applications, you are safer to confounding in your association analysis. Similarly, following a similar argument, you can say that it's very rare that the phenotype, the trait you are studying, will have a reverse causal relationship. So once you have an association, it could be direct or indirect, but usually it's never going to be reversely caused by your outcome. So sometimes it will help your interpretation. So there could be an argument to support this. But in addition to that, most existing genetic studies actually assume that the genes and the environments are independently affecting the phenotype. There's no interaction, it's just completely additive. This is really wishful thinking, and it's just oversimplification of the reality, as we'll see later using many different examples. There's actually not a very good example to support that, but just for some reason, in human genetics, the entire field rarely look at gene by environment interaction, and people sort of just treat environment as a nuisance parameter and don't want to talk about it. And to be fair, I used to be like this. I rarely thought about environment in our genetic studies, and this is really a shout out to our excellent collaborations here at UW Medicine, which is an initiative on social science genomics. We have a very collaborative group, including people from sociology or policy school and psychology. And in social sciences, environment sometimes is the question. You really can't ignore environment. And because of this collaboration, it really changed our perspectives and started thinking about pretty new questions that fewer people have been exploring in human genetics. And these are just 2 recent papers that we actually, Yu Chang wrote, back-to-back PNAS publications. This is a talk I gave a couple of weeks ago at the Center for Genomic Science Innovation. This is a different talk, but just very quickly, this is on the same realm of talking about how do we better account for environment in human genetic studies. On the left-hand side, again, you can't see the mouse, but I'll try to describe it. You can see this trio plot, right? The basic idea is, in a standard GWAS, what you do is you have someone's phenotype, you have someone's genotype. You have a large sample, you're trying to do regression, right? You're regressing your own phenotype on your own genotype. If you identify association, people try to think that maybe this is causal. Maybe there's some mechanism behind it. But what we realized is really, just because your DNA is associated with your phenotype, it doesn't necessarily mean that it is directly affecting your phenotype. I'll give you one example. For example, people have done big GWAS for educational attainment. So you can actually show that your DNA can predict, for example, college completion rate. But a lot of that could be because you have the genetic variant, you have the genes. It affects you somehow, maybe biologically, maybe in other ways, so that you can achieve a better education. But potentially another big part of it is actually you got the genetic variants from your parents, and your parents also have these variants. That could shape the parenting, shape the family environment, shape the environment you get as a kid, and that could causally shape your education. So that just because your DNA can predict education, we have empirical evidence that substantially part of that is because pretty much your DNA can predict your parents' DNA. It can predict what kind of family you're born into, and that's actually mediated through family environment. If you think the genetics of education is somewhat more too provocative, this is not by any means specific to education. You can think of, for example, if you talk about genetics of birth weight, fetus DNA can contribute to his or her own birth weight, but you can imagine mother's DNA will also contribute to the in utero environment that shape the baby's birth weight. So if you do a standard genome-wide association study by regressing everybody's birth weight on their own genotype, you get a mixture of two different kind of effects. So Yishan was able to build models to sort of dissect that. A lot of GWAS associations for special type of contact traits are actually confounded by this parental genotype, which is something that most people ignore in existing studies. Parents' genotype causally shape your genotype. Parents' genotype causally shape your phenotype in some way. And another example is on the right-hand side, which is a rare example where we demonstrate that the environment sometimes can actually causally shape the DNA variation in the population. Which I earlier said that this is pretty rare. Usually people don't think there's a reverse causation. But the story here is we actually did a GWAS. It's just a very straightforward GWAS design. But the phenotype is interesting. Is that in the UK we have 500,000 people. We can figure out the county and year of birth for everyone in that 500,000 people cohort. And then we can curate the infant mortality rate based on the year and county of birth, which is a quantity that summarizes basically how tough the environment is around the time this person is born. And there's a big variability across year and across regions in the UK. And if you do a GWAS on that, the idea is in some regions with bad environment, some people did not survive. The alleles disappeared from the population. In regions with lower infant mortality rate, this selection pressure is weaker. So if you are able to see genetic association with this environment, which is infant mortality rate, that actually gives you a sense about how the environment actually applies to the human population allele distribution and how the selection story works. And somewhat surprisingly, we actually are able to see very clean GWAS results. Just two lead loci coming out of this GWAS. One is for lactose intolerance. It's actually literally the gene LCT, which is lactase. Another is for bacterial infection. It's a resistance to bacterial infection and leprosy. And we can show that the selection pressure is, because we can do this year by year, the selection pressure actually peaks near 1942, which is exactly during the Blitz campaign in World War II in UK. So you can really demonstrate that even in modern contemporary human society, you still see this ongoing, well, at least within the past century, this ongoing selection pressure that gives you very specific selection signals. We like the story very much, but the point is, in most human genetic studies, people completely ignore environment. Here I'm trying to give you two examples. One is that your GWAS association can be completely mediated by family environment. In another case, the environment actually can causally change the DNA in the population. If you do not account for these factors in your study, it depends on the phenotype you want to study, but in some cases, it will completely ruin your interpretation. I'll pause a little, because see if people have questions, but if not, I can move forward. Okay, great. Today I want to talk about gene by environment interaction analysis. So it's interaction. It's a continuation to the talk that I gave at CGSI a couple of weeks ago. I know a number of you were there, so I'm not going to repeat the same stuff. Everything I'm going to say later is work by Jiachen Miao, who is a second-year PhD student in our own biomedical data science program. I don't have to tell you how good the work is. You'll find out very soon. I'm really impressed. And as I said earlier, I'm going to focus on one project, give you some details, talk about intuition, then talk about some ongoing work, which we find very exciting. So gene by environment interaction, I used to think it's extremely boring from a statistical standpoint, and just trying to be honest here. And I still think that the existing literature on method is pretty boring. Everything you see in the literature pretty much follows this generalized linear model you see on top. Which is that you have some sort of phenotype Y, you have a generalized linear model, right? On the left-hand side, you have the expectation, you have the link function. On the right-hand side, you have the intercept, you have the G main effect, so genetic main effect. You have the environment main effect, then you have an interaction term, right? So the genetic main effect is typically either a SNP or some kind of aggregated metric that summarizes information coming from a large number of SNPs. So more specifically, it could be a polygenic score, which I'll define just in a minute. And the environment could be, I'm talking about environment in a very broad sense. It could be a treatment. It could be a policy change. It could be birth cohort. It could be anything you want that is independent from genetics. In this talk, we're talking about, we assume that genes and environments are independent, which is kind of a big assumption. We think about this every day. But in the scope of my talk, I'm not going to, give you examples that really breaks this. And the parameter of interest is this beta I interaction coefficient. If you have a phenotype, if you define your G, if you define your E, it's fairly straightforward to just use a linear model and test whether G and E has an interaction. Whether beta I is 0 or not, you can get a p-value, you can get the effect size estimate. And just to make sure we're on the same page, when I said polygenic score earlier, it's shown on the right-hand side, actually. This is what I meant by it. It's essentially a weighted sum of allele counts. I is for ith individual, J is for J SNP, and each SNP allele count is weighted by a weight, Wj. And typically people use GWAS effect sizes, those regression coefficients in GWAS, to weight these SNPs. And it's essentially, when you do a GWAS, you quantify the effect size from each SNP on the phenotype, then you add them up. Calculate the Y hat for whatever phenotype Y you're trying to predict. That's what I meant by polygenic score. There are fancier methods to talk about how do you better estimate these weights given limited sample size, etc. But generally, this is the intuition. And like I said earlier, here in the scope of my talk, we assume gene and environment is independent, but we're not naive. We know this is a big assumption, and we have ways to deal with that in our analysis. But the perspective of understanding these associations are sometimes, interactions are sometimes different. For example, genetic epidemiologists will use this type of model to try to find an environmental factor that explains the heterogeneity of G effects, right? This is called, the technical jargon is incomplete penetrance. So some genetic variant will have a strong effect on disease outcome, but somehow in some people, it doesn't show as much strong association. In a different subgroup, it shows very strong association. What explains this differential genetic effect size? Could that be explained by environment? That's what people are interested in. But social scientists look at this in a very different perspective, although the model looks exactly the same. Or clinical researchers, actually, people have a pre-specified E in mind. Let's say I have a treatment. It's a randomized clinical trial. I have a treatment. But somehow it works on some people. It doesn't work on other people. Can I use genetics to define these subgroups, right? Is it possible that your genes modify the effect of my treatment? And in social science, for example, I have a major policy change. Right? I have cigarette taxation. Increase the tax of cigarette, make it more expensive. The goal is to reduce teenage smoking. But then turns out it works on some people, but it doesn't work on people, for example, who are really, really addicted to nicotine. And that could be indicated by a genetic marker that shows that this person is highly addicted to nicotine. So if this person is highly addicted, no matter how expensive you make cigarette, they're going to continue to smoke. So basically, you have differential effect size of the policy change, and people are interested in quantifying how that's really modified by genetic variants. in the context of using a polygenetic score to test interaction, you can imagine this being, if you can come up with a metric that summarizes contribution of 100, 200 different variants that quantify their genetic propensity of nicotine dependence, you can imagine that this G could potentially modify the effect of a policy like cigarette taxation. Yeah? So the study design in the field for this gene-by-environment interaction really is just, we started with candidate candidate G by E, which is that we hypothesize, for some reason, based on literature, based on our own experiment, these variants are interesting. We sort of know what they are doing, so we have a small list of variants, and then test whether or not they interact with another pre-specified environmental factor and get the p-value. And after a while, people really figure out this is not the right way to do things. So you get too high false positive rate. It's just the literature is just full of publication bias, and results that cannot be reproduced. Then people started doing hypothesis-free scan of SNP by interaction. So basically, do a similar thing, testing interaction between a particular SNP and the environmental factor, but do it for 10 million variants without specifying which variant are more interesting. And sort of like doing a GWAS of G by E interaction. But then very quickly realized that this is no hope because sample size is typically limited and you have 10 million independent tests, then you basically don't have any statistical power to identify a tiny bit of interaction effect. So then, which is why people started to do this polygenic score-based interaction. Is it possible that I have an external GWAS, let's say for nicotine dependence? Based on this GWAS, I get all these WJ on the top right-hand side. And then I can construct this polygenic score to quantify the propensity for nicotine dependence in my smaller cohort, which is defined as G in my model, and then test if that polygenic score G interacts with cigarette taxation in my study. Can I get a significant interaction? That's sort of the history and how 90% of the papers in G by E literature looks like. The reason why I said it's boring, it's a generalized linear model with an interaction coefficient. I don't see why that could be interesting. Then, in this talk, I'm going to try to rebuttal myself in a way by thinking about what interesting things could we possibly see from a method standpoint and improve the design of G by E studies. The question one, the first question I'm going to talk about is essentially, can we find better G? As I said earlier, most people just use a particular SNP or do this genome-widely, use 10 million SNPs to test interaction, or use a standard polygenetic score. Is it possible that we can better prioritize SNPs to find the genetic variants that are potentially more reactive to the environment, that are more likely to actually give you G by E result without looking at your data? And the second, is it possible to construct these better polygenetic scores that's different from a standard polygenetic score, but somehow quantifies the correct genetic propensity that is supposed to interact with environment? That's the point of the first question, or the project I'm about to talk about. Which leads us to this jargon, VQTL mapping. I'll explain what it is in a second. The idea is, what we are trying to see is, well, I'll use a motivation example first. So basically, there are two different kinds of flowers. You have dandelions, which, is pretty robust. No matter how good or bad the environment is, it can survive. It always looks the same. It's not a beautiful flower. But on average, let's say you have appearance as a quantitative measurement. On average, it's okay. But you also have orchid, which is a very different flower. It's it looks very luxurious. It looks beautiful when the environment is good, when you have the right amount of water and temperature. But it couldn't even survive if the environment is bad. So what we're trying to talk about here is it possible to find genetic variations that distinguishes orchids from dandelion? The point is, if you have gene by environment interaction, for example, genes defines two different flowers here, right? Environment has a big impact on orchid, but doesn't have as much impact on dandelion. So this is a gene by environment interaction. If you have gene by environment interaction, you are supposed to observe differential phenotypic variance between these groups defined by genetic variants. The idea is that phenotypic variance in orchid is supposed to be greater than the phenotypic variance of dandelion. And we are trying to quantify that. So statistically, this is trying to say that, as you can see in the middle panel, on the upper one, you can see that this illustration of gene by environment interaction for a particular SNP, we have 3 different genotypes, for example. And the three colors actually indicates three different environments. You can see that the genotype-phenotype association, which is quantified by the slope, is different between different environmental categories. But then if you then remove the color from this figure, what you will see is actually association between genotype categories, on the lower panel, by the way, and the phenotypic variation. Actually, not necessarily the mean, but we're trying to quantify how genotype will contribute to the variability of this quantitative trait. There are some models in the literature that handles this. For example, on the right-hand side, this paper by Alex Young and Peter Donnelly introduced this model called HLMM. I think it's heteroskedastic linear mixed model. The idea is really just, let's assume a normal distribution, but instead of only having a mean effect, right, you can see the expectation of this normal distribution is the function of G. G is your SNP genotype. Instead of having that, in addition to having that, you also have this, variance term. Basically, the log variance of this normal distribution is also a linear function of your genotype G. So then, this slope in that regression problem actually quantifies how genotype contribute to the phenotypic variability of this, distribution. Has been quite successful. I actually like the paper a lot. And there are a number of other tests that can help you. For example, just test, you have two different genotype groups. Can you figure out whether the variance of the phenotype is different? These methods generally have the limitation that they cannot adjust for covariates. It's just two-sample testing problem. It doesn't really produce this effect size, which will be helpful in downstream analysis. But there are a number of options you can potentially use here. And the limitation of models like HLMM, it has a very strong distribution assumption. If your underlying distribution is not normal, everything looks completely different. And it's not clear whether this type of likelihood-based method is actually robust to the distribution assumptions. And I'm going to show you examples to show that in practice this is actually quite important. Anyway, this is the intuition. We're trying to find VQTR. What is VQTR? It's genetic variance associated with trait variability. That's what V stands for. Then we have our idea. It's work by Jia Chen. We have a preprint. The idea is to use quantile regression to identify genetic variance associated with the phenotypic variability. You can see on the left-hand side, if the genetic category or genetic variant genotype is only associated with the mean of the phenotype, as you can see in the left, but on the upper panel, then if you fit quantile regression based on different quantiles, let's say you have a quantile regression based on 20% quantile and then 80% quantile, and then maybe median, you should see that the slope of these quantile regressions based on different quantile taus to be the same. These lines should be parallel. If this genetic variant is actually contributing to the phenotypic variability, then by doing a similar analysis, you should expect to see the slope to actually differ. The 20% quantile regression should give you a very different effect size compared to an 80% quantile regression. That's a general intuition. So then initially we had this intuition, and what Jia Chen tried in the beginning was that how about we just fix the quantiles to be 25% versus 75%, and then compare the slope, right? So we get the sense of this sort of the interquartile range of this, beta, this slope difference. But then it turns out to work okay, but the obvious question is why only 25% and 75%? How do you really combine this information across more quantiles? So that's really illustrated by the sort of formula on the very right-hand side. Eventually, we wanted to build something that looks like this. So for many different tau, which is a quantile, we want to quantify the difference between a pair of regression coefficients. The intuition is we want to compare 90% quantile regression slope versus 10% quantile regression slope. Similarly, compare 80 versus 20, 75 versus 25. Then add up all these differences so that we are actually leveraging information across the entire distribution of this quantile and summarize that into this metric. We call it beta QI quantile integral. And then if you can estimate this, then when this is 0, it gives you a strong evidence that these quantile regression slopes are actually identical across all the tau you're talking about. Sounds good. Empirically, how do we actually make it happen? In practice, though, we're not taking an integral. On the right-hand side, you see it's an integral. In practice, you just need to select a grid of quantile and use this step function to approximate this quantity, actually. But to be able to do this, you need to have a large K, as you see in the first formula. K is the number of quantiles you need in this grid. Then basically, if k is 1,000, then you basically need to fit, say, 2,000 different quantile regression for each genetic variant. That's virtually impossible. And I'll come back to this just later on the same slide. But we actually know how to fit quantile regression. In the middle, you can see this quantile regression linear model, which is just say that in a standard linear regression, on the left-hand side, you have conditional expectation. Here is a conditional quantile based on a quantile tau. This quantile is a linear function of genotype G and also covariance C, right? The parameter of interest is this beta tau k. And then to fit this regression, it's just basically you need to change the loss function from L2 distance to this specifically designed loss for quantile regression. And the special case is that if you want to fit a median regression using 50% quantile, this is essentially the L1 norm. It's actually very similar to a least square estimate. Then the point is, yes, if you give me a quantile, I can fit a quantile regression, but I need to do it 2,000 times for each SNP, and I have 10 million SNPs. And each quantile regression involves minimization of this loss, so it's an iterative approach, so it's virtually impossible. If we want to do this in a GWAS setting, we have to fit 20 billion quantile regressions. So that's a big computational challenge. And additionally, even if you are able to construct this beta hat QI as shown on the very top of the slide, it's actually not a trivial problem to calculate the standard error of this point estimate, because it's a linear combination of many different quantile regression estimates. They're not independent. It's not a trivial problem to characterize the covariance structure between these estimates. So that's obviously just impractical. So the idea is intuitive, but it's impractical. But the good news is Jiaxin figured it out. I'll tell you what we do and eventually try to give you some intuition about what this is about. So the idea is basically you need to do it in several different steps. The first step is that you basically need to fit a quantile regression for all the quantiles you're interested in, but by regressing the phenotype on covariates only. There's no genetics in it. After you regress out the the covariates, you can construct this in the second bullet. You can construct this so-called quantile rank scores. What I'm going to say here is just that the A hat can be directly calculated based on results in the first step, and then N is just sample size, tau is the quantile you're interested in. Then you need to figure out how to calculate this term SE of gamma hat. Gamma is regression coefficient in the following formula. It'll be easier if you can see the mouse. But basically, what we are doing is you need to fit another quantile regression. Again, covariates C. But then you add a randomly sampled variable from standard normal distribution into it, which is D. And the regression coefficient of that D term is gamma. And you just randomly sample a variable, throw it into the quantile regression, and it will give you a standard error estimate of the gamma. Use that back in the second step, then you can construct this quantile rank scores Y. Why they're useful? Because we figured out if you have this Y, then eventually you can actually come up with almost equally good estimate of your quantile regression effect size between the SNP and the phenotype following the formula in the very bottom, which looks like a linear regression effect size. G here is basically the residualized genotype for that SNP, and Y tau is a quantile rank score you construct in the second bullet. Then you can basically do ordinary least square by regressing this new phenotype, Y, on your genotype SNP. It's sort of like doing a GWAS. So we have very efficient computational tools to do this. And Jiaxin figured out the proof. You can actually show that this new estimate is very similar to what you would get following this sophisticated iterative approach that minimizes this specially designed loss. But the difference is, to be able to do this, you don't have to fit 2,000 quantile regressions for each SNP. What you need to do is to fit 2,000 quantile regressions together for the covariates. You don't have 10 million multiplier on it, and then that's basically done. Then you can construct the new phenotype and run another GWAS, which is computationally very efficient. And we have very good tools to deal with that. So hopefully this is intuitive enough, but not necessarily intuitive, but at least you understand what we're doing. So I'm going to give you a little bit more intuition of what exactly we're doing here. The idea is, in the second step, we're trying to develop this quantile rank score. The idea is basically Jiaxin dug up this ancient text, by that I mean paper from the 70s, maybe not that ancient. But the idea is you can have a very good approximate of the p-value or the test statistics of quantile regression. if you dichotomize your phenotype. Right? Like in quantum regression, it's a quantitative phenotype. You're trying to fit, let's say, a median regression. You assume the median of this distribution is a linear function of the genotype. You're trying to estimate the slope, right? But there's an alternative way to do this if you're only interested in p-value, which is you dichotomize the phenotype into 1 or -1. It's 1 if your phenotype Y is above the median across samples. It's -1 if your phenotype Y is below the median across all samples. If you dichotomize the continuous phenotype into this binary outcome, you can just run a linear regression on this binary outcome and regress it on the SNP. And the linear regression will give you a very similar test statistic compared to the actual quantile regression test statistic. So then, but only having test statistics is not good enough. You have to sort of balance it a bit using the standard error of an actual quantile regression coefficient, so that you can actually infer the effect size, right? We're not just interested in p-value, but in our case, very fortunately, we can show that by using this randomly sampled variable from standard normal distribution, you can essentially approximate this standard error of beta hat you actually need from quantile regression. The reason for this approximation to work is essentially what I said in the beginning, it's homogeneity. Because every single variance has virtually non-existent effect size on the phenotype, so the actual effect size we're talking about is tiny. So if you actually sample a 0 effect, which is completely random from anything, add it into a quantile regression, the standard error you get is a good approximate of the real standard error you would have gotten if you fit the quantile regression using a real genotype data. So that's all the intuition. Hopefully, it's kind of hard to do it on Zoom, but hopefully people get the idea. But I'll pause a little bit to see if I have questions. Good, great. Then if you're able to do this, then just a reminder, eventually you want to get this linear combination of different slopes based on different quantile taus. But then to achieve this, it's very straightforward. You just similarly combine the quantile score phenotypes you constructed on the previous slide, and then eventually have a more composite score, just run linear regression, do ordinary square, and the regression coefficient is exactly what you need. This is only slightly slower than typical GWAS. It's fairly easy to implement. This is why this is methodologically interesting. And you can actually compare the regression coefficient you get using our approach, which is on the y-axis, versus a standard regression coefficient you would have gotten using a traditional quantile regression approach. You can see that it's highly consistent. The correlation is 0.99. So it really doesn't, it really actually approximates the effect size quite well. By the way, the method is called QUAIL. It's called quantile integral linear model. We did some simulations. Simulations always work. But the idea is that, when you're right about the model assumption, everything is normal and nice. No existing method will have type error inflation, as seen in the panel A. But if you're wrong about the underlying assumption, let's say it's just slightly different, right? The actual distribution is T distribution, it's not normal. Then those models that assumes the normality assumption breaks very easily. Immediately you get highly inflated type error, as you will see in panels B and C. But then in terms of power, across different distribution settings, the QUAIL approach seems to, in some cases, either give you a consistent result or outperform depending on what distribution you choose. And then our method is also able to adjust for covariates. Just to remind you that this is not the effect of covariates on the mean of the distribution. Basically, we are saying we can actually adjust for the potential confounding effect of covariates on the variability of the distribution. So several existing methods couldn't do it. That's what panel G is about. So you get type error inflation if you don't adjust for confounding. Then in real data analysis, you can actually do this VQTL mapping for BMI as the outcome in UK Biobank. You can see that, by the way, you can also do the SNP by E interaction. Yeah.
There's a question.
Oh, yeah, go ahead.
Michael asked, you need the subject-specific responses, not summary stats for QUAL, right?
That is correct, yes. For this, we actually need individual level data. Oh, yeah, the question is, I guess people actually can see it, but the question is, do we actually need individual level data for this QUAIL approach, and not just summary statistics? And that is correct, yes. We need individual level data to fit QUAIL to construct the quantile integral phenotype and then run GWAS. But after doing this, you can get the summary statistic for QUAIL. That can be used in downstream application in different ways. Yeah. Okay, good. Then this is just to show that the VQTL we identified in this approach are actually strongly enriched for SNP by E interaction signals. We did an actual SNP by E interaction scan in the UK Biobank using BMI as the outcome and sedentary behavior, which is a metric that quantifies how many hours you sit every day, how many hours do you drive, how many hours do you use laptop, that sort of thing. And that interaction with SNP. We did a SNP by sedentary behavior interaction scan for 10 million SNPs in UK Biobank and identify some top signals with G by E signals. You can see that the top VQTL are much more strongly enriched for these interaction signals compared to the top signals you would get from a standard GWAS for BMI. On the left-hand side, you have this differential fold enrichment. You can see the pink bars are always taller. On the right-hand side, it's a similar thing. It's just change the metric into p-value of enrichment. You can see that the enrichment in VQTL is always stronger. And then you can also construct a variance-based polygenic score for basically the phenotypic variability. Just like the PGS that people typically use, but instead of using a GWAS effect size as the weight, we're using the Quayle effect size, right? So then you aggregate the contribution coming from a lot of different SNPs, then the score you get will actually quantify the phenotypic variability. Then on the lower right-hand side, you can see that across 3 different cohorts, you can consistently see that this score is significantly predictive of the phenotypic variability. For example, in panel A, in the middle, this is the sort of the between-person variability. Basically, if you stratify the population, each cohort based on 5 different quintiles of your variance PGS, you can see that in the lowest quintile, the phenotypic variability is much lower. In the highest quintile, the variance is much higher. You can see this linear trend. And then, in addition to that, we can even predict within individual variability. So we build a model with the lower left corner, this figure in mind, which is that, okay, so if you partition individual based on different genotype, maybe the phenotypic variance will be different across groups. But another interesting metric we're interested in looking at is actually for a particular individual, if this person has a high, say, variance PGS, over time, because BMI is something that's more dynamic, right? Do we actually see more fluctuation of this person's BMI given the very high variance PGS? Would this person react to environment more sensitively somehow? And we are also able to see that predictive power. And we compare that Quayle VPGS versus existing approaches, we can always see that it's the most significant predictive of both population level variability and within individual variability in 3 independent cohorts. So it's actually quite effective. I wrap up this project by talking about, just by giving you a quick summary. Again, the question is, can we actually find better G? Yes. So basically, we can do a VQTL mapping by identifying genetic variants that are associated with phenotypic variance, not just the mean. And turns out those variants are more strongly enriched for gene-environment interactions. You can also aggregate this effect across many different markers and produce this variance PGS and replace the standard polygenic score into this variance PGS. You can also show that it gives you a stronger interaction with environmental factors. We have a paper with Lauren Schmitz that exactly showed that, and we have ongoing projects to explore further applications of this. This is what I have to say about the first project. I'll stop for 5-10 seconds, see if people have questions. If not, the next question is the exact same model, but the question is, can we actually find better ways to estimate beta I? Because that is a regression coefficient that people are actually interested in. And in this specific project, I'm talking about a PGS by E interaction study, which is 80% of all existing literature. You need a GWAS for a phenotype. Based on that GWAS, you can construct a polygenic score for that phenotype in your study, which is your G. And then you can test whether G and the pre-specified environment has an interaction effect quantified by beta I. That's generally the design. It's also illustrated in the lower half of this figure. Basically, on the left-hand side, you have to run a GWAS in a cohort, produce GWAS summary statistics. And then on the lower right-hand side, it shows that based on the summary statistics, you can produce polygenic score. And in your cohort, you can test PGS by E and estimate alpha I. My problem with this is, this study design actually requires 2 different cohorts that are independent. For the PGS to be usable in your analysis cohort, it needs to come from a completely independent GWAS. And B, polygenic score is known to have terrible prediction accuracy. Yes, it is non-zero prediction power, but it is not highly predictive. When you construct these polygenic scores, you actually lose a lot of information. The kind of beta I you're interested in estimating is really the interaction between this true polygenic score, when your external GWAS has infinite sample size, and then how that interacts with your environment in your study. right? So then basically we're trying to get this beta I when the GWAS sample size goes to infinity without an actual GWAS with infinite sample size. That's our goal here. Is it possible to get an unbiased estimate of beta I for this Oracle PGS that is difficult to quantify without actually having to calculate the PGS itself? Turns out we can, and I'm going to step back and use 2 slides to talk about something that looks completely different, and I promise I'll drag you back very soon to talk about GMI environment interaction. And the problem I want to talk about is genetic correlation estimation. It's a topic that I enjoy very much. Looks completely different. The idea is this is a metric that quantifies the genetic similarity of 2 different traits. You can say you have a GWAS for schizophrenia, you have a GWAS for bipolar disorder. How much genetic sharing are you talking about? To what extent does genetics contribute to both phenotypes? Typical model looks like this. So basically, you have 2 linear models. Sorry, you can't see the mouse, but you have a model for Y1, you have a model for Y2, just linear models. X and Z are N by P matrices, right? You have N individuals and P SNPs, and beta and gamma are the effect sizes of these SNPs on phenotype 1 and phenotype 2. And people use the random effect models. Beta and gamma actually are random effects, and they have a covariance, which is to say that every single SNP has 2 effects, one on schizophrenia, one on bipolar. These 2 effects are random variables that have a covariance. And I want to estimate this parameter that's shared across 10 million SNPs. And basically, this is what we mean by genetic covariance or genetic correlation. This is a metric that quantifies how similar the effect size is between 2 different phenotypes. And this goes back to Michael's comment earlier. You can actually use GWAS summary statistics to estimate this. The intuition is if you have a GWAS for schizophrenia, you have a long vector of SNP effect sizes. Similarly, you have a GWAS for bipolar disorder, you have a long vector for SNP effect sizes. You can fit, for example, this LD score regression approach, which is a paper, it's a fantastic paper, and regress the product of these test statistics Z-scores on LD score, and the slope turns out to be exactly the parameter, the covariance parameter you want to estimate. And another very nice feature of genetic correlation analysis is, let's say your GWAS for schizophrenia and bipolar are actually not independent. In a lot of cases, they actually share samples. For example, they can share controls, or for quantitative trait, they can have a study conducted in UK Biobank for height, but another study conducted in the same cohort for blood pressure. When you test genetic correlation, the study samples are completely dependent. We are talking about the same cohort. LD score regression, or this type of genetic correlation estimation approach, is actually robust to that. The idea is when you do the similar regression, when you look at the formula at the very lower bottom of this slide, when you do a regression by regressing the product of test statistics on LD score, LJ, all these bias introduced because of sample overlap will be on the intercept of this regression. The slope is still a very good estimate of the true parameter you're actually interested in. So it's incredibly robust. It's not our paper, but it's one of my favorite papers in the past few years. This is a background. I'll give you one example. I think it's a fun example because people actually did this for COVID genetics. You can do COVID genetics for reported infection. It's basically testing positive versus negative, or for hospitalization, so more severe cases, and then test genetic correlation with everything out there. You can see that whether you get COVID or not, whether you test positive or not, really there's a very strong genetic correlation with risk tolerance. The higher tolerance you have to risks, maybe it quantifies the kind of behavior you're going to be engaged in, and then you have a higher risk for getting COVID. Okay? But when you look at hospitalization, when you look at once you get COVID, do you actually need hospitalization? Do you need critical care? The signal is completely not there. In that case, what matters more is on the right-hand side is actually these clinical risk factors like stroke, lupus, etc. So immunity. So, but that has nothing to do with how likely you're going to get COVID. It's only genetically correlated with the severity of the disease. I think it's a very cool example to showcase this idea. However, you can see the power of this type of approach because you only have GWAS summary set for COVID subtypes, and everything else only also is GWAS summary statistics. You can test all these correlations without having all this data to be measured on a single cohort. So it's a very useful and robust method for you to be able to say anything. I'll skip some details, but I just want to say that we actually have very good expertise on this topic. It's one of the specialties in my group. So we are quite familiar. We think about genetic correlation estimation every week. And so we're familiar with context, which is why we realized this could be a useful tool to actually quantify gene-by-environment interaction. Long story short, you can see in the first formula, in the middle of the slide, quantitative phenotype Y on the left-hand side, then you have genotype main effect, beta G. You have the environment main effect, beta E. You have the interaction effect, beta I. Using a very similar model, what we're trying to say is the ultimate interaction coefficient you're interested in estimating, which is the interaction between the oracle PGS and the environment, is exactly the genetic correlation between beta G main effect and beta I interaction effect. Of course, you have some constant involved in this. I'm not showing you all the details. But basically, if you are able to quantify the genetic correlation between beta G and beta I, both of which are very long vectors, right? Very long vector because they have many, many different SNPs, then you can solve the problem. It's going to be an unbiased estimate of that alpha I interaction coefficient we're interested in testing in the beginning. You do not need the external GWAS to build this not so accurate polygenic risk score. And you can also have sample overlap between the GWAS you use to estimate main effect and also the GWAS used to estimate interaction. To better illustrate this, this is sort of the design here. To be able to do this, by the way, the method is called Pigeon. That's similar to Quail, have a bird suite of methods, polygenic gene environment interaction. So what you need to do is you need to run a GWAS in some cohort, which estimates SNP main effect on A phenotype. On the right-hand side, you need to run a SNP by interaction scan for many, many different SNPs. Just like GWAS, but then you report the interaction coefficient and corresponding p-values and get a SNP by E summary statistics. Then all you need to do is to test the genetic correlation between these two. And the parameter will give you exactly what you need in that PGS by E interaction model. And the two cohorts you use to run GWAS and SNP by E interaction can have arbitrary sample overlap. We're about to run out of time, so I'll try to wrap up. And some very basic simulations, when there's no sample overlap, the story is no method has type error inflation, as shown in panel B here. But as the true effect size increases, methods sort of perform similarly. Our approach has a similar statistical power compared to the best polygenic score out there. The orange one is actually the oracle PGS, which nobody has. If you know the true PGS, you can do better, but nobody has that. And on the right-hand side, it actually shows the parameter estimate. You can see the green bar, which is our results. The green box is always unbiased. But if you use the PGS to estimate this interaction coefficient, because your PGS is so noisy, you're not getting the true PGS. The purple and the blue boxes are always below the true parameters, so you get underestimated effect sizes of interaction. When there's full sample overlap, in panel E, you'll see that PGS method is completely useless. You get total inflation of type error even if the x-axis is 0, so there's no interaction anyway. This is totally driven by sample overlap and artifact. On the right-hand side, you actually get overestimated PGS by E effect using PGS approach, but our approach remains robust, even if there is a complete sample overlap. So just simulations. I'll skip this example and give you one real data. Basically, we are doing this for SNP by sex interaction for 530 traits in UK biobank using a sample size of 500,000. The idea is that your phenotype outcome can be 500 traits, can be everything. And we're trying to test 50 polygenic risk scores interaction with biological sex. And see, suppose there is a sex-specific genetic architecture for some trait Y, what could explain these sex differences of the genetic basis of that phenotype Y? So we're interested in testing whether PGS and E have an interaction, beta I here. But we are doing this using our approach. And it's virtually impossible to do this using PGS approach because almost all GWAS out there use UK biobank samples. If you want to do this in UK biobank, there's a big sample overlap. You can avoid that, but our approach is robust to that. One interesting example that I'll talk about today is we did this for BMI, which is to say that the genetic basis for BMI looks different between males and females. We're trying to find the reason why it's different. Looks like the strongest PGS interactor is actually anorexia. The interaction is actually the anorexia polygenic score is less predictive of BMI in males, much, much less predictive in males than in females. So that partially explains why if you do GWAS on BMI in males and females, it looks somewhat different. And this is a very good way to identify what is actually explaining that. And you can do this in a hypothesis-free scan because you can just run a SNP by sex interaction analysis on BMI and get the summary stats and test its correlation with 50 other traits, anorexia pops up as the most significant result. It's even more significant than BMI itself. So we think this story is quite convincing and we like it very much. I'll wrap up. Basically, I talked about two different projects, both of which are about gene-by-environment interaction. Queer is more about, can we find better G? Can we prioritize SNP better by looking at the SNP that's contributing to phenotypic variability? Or can we construct this variance PGS that quantifies how plastic you are, how sensitive you are for environment, how likely you're going to react to environmental variability, and show different phenotype. And then PGen is a very different statistical framework to estimate this PGS by interaction coefficient. It has a number of advantages. It produces unbiased estimates, even if your external GWAS has limited sample size, and it's it doesn't. It's also robust to sample overlap, and it's also easy to implement meta-analysis, which I'm not going to get into, but you already get the idea. So I'll stop here. All this work is done by Jia Chen, and Lauren and Jason, our collaborators in Initiative on Social Genomics, have contributed very substantially to this project as well. And Gefei is undergrads who has helped perform a lot of analysis. I'll stop here and see if people have questions. Thank you very much.
Okay, thank you, Cheng-Shi, for this very nice presentation. And also, thanks for the audience from both Zoom and also in the auditorium here. And thanks to Chris Harrison and his colleagues who are working very hard to make everything smooth and feasible. Any questions, you can feel free to speak.
Hopefully that's because I was clear.
Yeah, there's a question here. So let me repeat. So the question is, what do you see as the hurdles and opportunities to using PGS in clinical setting?
I don't see any major hurdles. If your samples have genome-wide genetic data, it's fairly straightforward to compute polygenic risk score for thousands of phenotypes based on published studies. The question is just you don't necessarily know which score you should use. What will actually explain, for example, the heterogeneity of your treatment effect? So you have to involve some sort of hypothesis-free scan by trying different scores that could hurt your power, right? But that's a pretty standard, statistical problem, and we have tools to deal with that. And I would assume that these studies typically have a lower sample size compared to the GWAS we're talking about. So then you may not have sufficient sample size to identify these interactions if the interaction effect size is not strong enough. It's a very empirical question, right? So if the interaction is actually very big, a particular score explains the treatment heterogeneity, then maybe you're hitting the jackpot. But in practice, if you only have, 100 samples, it will be underpowered.
Okay, thank you. There's another question, so let me repeat. Any suggestion to use PGS for the prospective design? Is it possible to use the information during the trials?
It is possible. Basically, the good news is DNA variation doesn't change after you're born. So you can use this to quantify the genetic propensity for pretty much whatever you want, even if you do not directly measure those phenotypes, right? So for example, you can quantify people's Alzheimer's risk, blood pressure, hypertension. If you think to have a genetic propensity for these factors can help you prioritize samples to decide who to exclude or include in your study, then this will be very useful. And all you need is to do genome-wide array for all the samples you have, and it's pretty cheap these days. It's $50 per sample. I don't think it's being actively used, and Tom can correct me if I'm wrong, in the field, but I don't necessarily see this as any issue. It's just people may not be very familiar with these techniques, and we need to talk more between different sub-disciplines in this type of data science problems.
Okay, thank you. Actually, we are already running out of time, so if you have other commitments, feel free to leave. And if you want to speak to Cheng-Shi, even you are at Zoom, just speak. I can test whether he can hear you. And also, if you see any problems about the connection issues, and also somewhere that we can improve in the next few times, let me know by sending me an email or directly. Okay. Thank you, everyone.
Thank you. I'll stay here for a little longer to see if there are questions. And still, thank you for attending.
That's great.
Yeah. Still a little weird to do it hybrid, huh?
Yeah, I think the most difficult part is the interaction with the Zoom.
That's right.